really excited on the behalf of bioengineering to be introducing our 4 brand new faculty to the department. And with them, they bring 4 new directions for research in the department. And this adds to our strength in our 6 general research areas that we have in bioengineering. So the intent in this talk today is that their research works as what we call in a format as a research short format. So that is they're going to present what essentially is a preview to what they do in 12 minutes each. So the way I want to set this up is I'm going to be introducing all of them together, and then they're going to go in the order, alphabetical order, last name. So that's going to be Yichen, Jacopo, Carolyn, and Girgis. So they're going to go in that order. And after 12 minutes each, then we open it up for questions for everybody. So I'd ask you all to hold your questions to the end. And the intent here is you might have a lot more conversations with them outside of this. So this is the first introduction to all of you. For the students, these are new areas of research that are there in the department. So the idea here is it's going to facilitate collaborations with the UT Southwestern, with other departments in natural sciences and mathematics, as well as in the Johnson School and the behavioral and brain sciences. So this is our intent behind introducing our faculty through this virtual format. So thank you all for coming and thank you for your attention. So let me start with the introductions. So first is Yichen. So Dr. Yichen Ding is an assistant professor of bioengineering at the University of Texas at Dallas. And prior to joining UT Dallas in August 2020, he graduated with his BS in optomechanical engineering from Tsinghua University and his PhD in biomedical engineering from Peking University. He completed his postdoctoral training in the School of Medicine and Engineering at UCLA in 2020. He is the recipient of the NIH R00 Award, and he's a co-author of more than 40 peer-reviewed journal publications. And so he's going to be talking in the areas of optical imaging, computational methods pertinent to cardiac injury, and with a special emphasis on ventricular imaging, machine learning, and virtual reality. The second speaker for today is Dr. Jacopo Ferruzzi. So Dr. Ferruzzi is again an assistant professor in the UT Dallas Bioengineering, and he's the principal investigator of the Tissue Mechanics and Remodeling Laboratory. His research expertise is in the areas of biomechanics and mechanobiology, and the goal of his research is to improve fundamental understanding of diseases associated with and driven by abnormal tissue mechanics. So he has extensive collaborations, and he's got his training from the University of Pisa, Texas A&M, and Yale University. And he's explored biomechanics in breast cancer by employing 3D tissue culture techniques, label-free imaging techniques, and he's building out a highly multidisciplinary research program here, bringing together mechanical properties, extracellular matrix organization, and cellular function. Our third speaker today is Dr. Caroline Jones, and she, of course, is an assistant professor here in the Department of Bioengineering. She came to us from the Department of Biological Sciences at Virginia Tech, where she was an assistant professor from 2015 to 2020. Her lab, again, is highly multidisciplinary, and it bridges both engineering as well as immunology, and her intent is to prevent, diagnose, and treat multiple immune-related disorders. And so towards this end, she has been working in designing innovative lab-on-a-chip platforms, developing imaging techniques towards tracking host cell pathogen interactions, and merging quantitative single-cell measurements and mathematical modeling. She's the recipient of the NIH R35 Award, which is also known as the MIRA Award. And again, she has authored over 30 scientific publications and the co-inventor of a patent. So welcome, Caroline. And our final speaker today is Dr. Girgis Obaid, and who also is an assistant professor here in the Department of Bioengineering. He has over 10 years of experience in tumor-targeted nanotechnology, and his expertise lies in the realm of molecular-targeted photodynamic-based therapy towards the goal of improving tumor delivery, reducing toxicity, and maximizing therapeutic benefits. So Dr. Obaid has started to build out his lab. He started with us early 2020, and he started to make wonderful collaborations with our research faculty at Southwestern, as well as here at UTD. He's again the recipient of the NIH R00 Award, and he's now bringing to UT Dallas and to our department the ability to use photonically activated nanostructures as a means for regulating drug delivery, treatment regimen design, and spatiotemporal induction of cancer therapy. So with that, welcome the four of you. And we all look forward to your various research talks. And Yichen, you go up first. The floor is yours. Thank you.
Thank you. Good afternoon, everyone. My name is Yichen Ding. I'm a new assistant professor in bioengineering. I'm a little bit excited to share with you about my research today. Actually, today's topic is advanced optical imaging and computational analysis for developmental cardiac mechanics. Before we get started, I would like to invite you to take a look at this virtual reality video. This is about adult zebrafish heart. So right now we are walking inside the atria of the heart. Besides visualization, actually we are also able to measure the distance between two valve leaflets like this. After that, actually we can also walk inside the ventricle inside in the heart. And right now we are inside the ventricle in zebrafish heart. All the red structures you are visualizing right now, we call that trabecular. This video was recorded when one of our collaborators was using our platform. We give them immersive feeling and user-directed microenvironment. In this kind of virtual reality environment, your perspective is not limited anymore. And also, this is just a prototype, but we believe in future, we are able to combine simulation and other analysis together in this kind of model. So that means we are able to provide or we are able to establish a virtual lab. So in future, we can use this kind of virtual lab to test our hypothesis for further studies. To make this happen, actually, we developed multiple methods. First, if you want to if you want to capture the heart, we need to build an imaging system. So that's why we call that light sheet fluorescent microscopy. And second, if you want a computer to recognize myocardium inside the heart, we developed the machine learning based method for image segmentation. We call that seg transform. Finally, we want to use virtual reality as a platform to provide this kind of interactive virtual lab. So that's why we want to combine imaging, machine learning, and virtual reality together for our further study. The reason why we want to do this is because our biological interest is cardiac injury and regeneration. As we know, heart failure is a global pandemic. It's another pandemic, actually, affecting at least 26 million people worldwide. No matter systolic heart failure or diastolic heart failure, it involves morphological change and functional change. Our human beings do not have the capability to repair or regenerate cardiomyocyte after it's injured. However, in zebrafish or neonatal mice, they have this capability to regenerate or repair the ventricle or repair the heart, even though you cut maybe 1/3 of the ventricle. However, in adult mice, they lose this capability as well. The underlying mechanism is still unknown, so that's why we want to use our engineering method to provide a new developmental and mechanical insight into this field. So the first step in our lab, we need to build an imaging system. We call that light sheet fluorescence microscopy. This is a cutting-edge imaging system. It allows us to capture morphological and functional changes in the heart. We build an imaging system from scratch, and we also push the limit of this system. We want rapid acquisition, we want higher spatial resolution, and also less photobleaching or phototoxicity. With this kind of imaging system, it allows us to capture image the whole neonatal mouse heart within 1 minute. And also, we don't need to mechanically slice the heart. The yellow part the yellow part you are visualizing right now is a ventricular cavity inside the heart. We combine our imaging method with optical clearing method together. Optical clearing method allows us to render the tissue or organ transparent. After you put your tissue or organs in the chemical solution, 1 or 2 weeks later, we are able to wash out the lipid. So that's why you are able to see the grids under or behind the heart. So this is for ex vivo study. But if we want to perform some in vivo study, we use embryonic zebrafish as an animal model. We are able to use our imaging system to capture the cardiac contraction as well as the blood flow from atrial to ventricle inside the heart. That means in future, we could study cardiac contractility and fluid dynamics together at the same time. So that's about our imaging system. The second part of our work machine learning-based image segmentation. We call that sec transform. Today, we do not have quite a lot of time to go over the details, but I want to highlight our method is a PCA-based method. PCA stands for Principal Component Analysis. It's different from neural network. Neural network is a deep learning method. In my words, actually, neural network is like a black box or traditional Chinese medicine. So most people don't know the details, but sometimes it works very well. However, PCA-based method, like modern medicine, so we want to know all the details at each step. That means we have to spend quite a lot of time at each step to design the architecture. But don't take me wrong, each method has its own advantage and disadvantage. The reason why we want to use a PCA-based method because it's proper for our application. So if you take a look at our dumples, here is our raw data of the 2D slice of the adult zebrafish heart. So we spent almost 20 hours to manually segment the myocardium in this single slice. But can you imagine if we have hundreds of slices like this? It's a labor-intensive work. So that's why we decided to use a machine learning or deep learning method for image segmentation. However, we realized if we are able to use a PCA-based method, we could generate more details when we use the same training images. In contrast to UNet. UNet is a very popular neural network deep learning method. So that's the reason why we want to develop this kind of sec transform-based machine learning method for image segmentation. The third part of my work is virtual reality-based interactive quantification. So we already developed a pipeline from imaging to virtual reality. As long as you follow our pipeline, you are able to download your digital model on your cell phone, and you just need to install this application on your cell phone and combine it with Google Cardboard or maybe some other virtual reality high-site. You are able to have those kind of immersive feeling as well as before. The reason why we want to do this because when I was a student, we have quite a lot of collaborators in the lab, but some of them just want us to provide the technical support. At that time, we realized why not establish a platform for user directly to use it. So that's why we developed this kind of virtual reality platform. So in future, we want to combine this virtual reality platform with our imaging method and also machine learning method together. We want to establish this kind of interactive virtual lab. So in future, we are able to perform simulation and further analysis in this kind of platform. Besides the first video I showed in the beginning, here is another one. It's about a contracting heart in zebrafish. It's also in the virtual reality mode, but in this video, actually we are also able to measure the volume change between different frames. This is just a prototype, but it makes us believe we are able to combine everything together to establish this kind of virtual lab in future for further analysis. To summarize my work, actually, we have 3 different aims. The first aim is about detection. That means we build a light sheet imaging system to capture the images. The second part is about segmentation. We developed a machine learning method named sec transform. Finally, we want to combine virtual reality with imaging and segmentation together to establish this kind of interactive virtual lab in future. Although our methods are established for cardiac studies, our imaging system is ready for neuroscience or material science as well. And our computational methods are also compatible with CT, MRI, or ultrasound images. So that gives us quite a lot of possibility for future work. Finally, I would like to acknowledge our collaborators and funding support. So I just started my lab at UTD, so I have several positions open right now. If you are interested, just feel free to send me an email at yichen.ding@utdallas.edu. Thank you so much. Thank you.
All right, well, thank you very much. And first of all, I would like to thank the department for the opportunity of presenting an overview of my research. And I'd like to start by stating that in biological tissues, structure relates dynamically to function. And this was first observed in the bone and led in 1870 to the formulation of what is known as Wolff's law, which states that bones grow and remodel throughout life to adapt to their mechanical environment. Now, over the past century and a half, we have moved from observing the macroscopic effects of tissue growth and remodeling to analyzing and investigating the mechanisms at the microscale. And specifically, now we know that tissues remodel via mutual interactions between the resident cells and their surrounding extracellular matrix. In particular, the ECM is more than a passive scaffold, and it's a complex network of proteins, glycoproteins, and glycosaminoglycans that not only regulates cell function, but also controls the tissue's mechanical properties. And in particular, for my work, I'm interested in two ECM proteins, being elastin and collagen, which control key features of tissue mechanics. Now, to just give you an idea of what actually physiological remodeling looks like, I like to show this some of the results of the NASA twin study, which reported how a year in space affects human biology and physiology. And among other findings, this study reports that changes in vascular structure and function develop early during spaceflight and persist throughout and after the mission. And in particular, as you can see here, these changes localized locally on the arterial wall, but also have global effects. And this local global connection is still quite unclear how it functions. And so this allows me to highlight some of the key questions that have characterized my research to date and will continue to characterize my future efforts. That is, what are the basic mechanisms of soft tissue remodeling? And how does local tissue remodeling impact overall physiology and pathology? And so to provide you an example with that, I'd like to talk briefly about the relationship between arterial stiffness and blood pressure. Now, stiffening of arteries, like the aorta, is an established consequence of hypertension. However, just a few years ago, it wasn't yet clear whether arterial stiffness could itself cause changes in blood pressure. And this is particularly relevant to normal aging. As we age, our arteries stiffen up and we develop hypertension. Now, part of this lack of understanding is caused by how these different metrics are measured. Arterial stiffness is measured locally by estimating the mechanics of the arterial wall, whereas blood pressure on the right is measured globally. And it usually involves a variety of hemodynamic metrics that involve the flow of blood. Now, during my PhD work, I proposed an integrated way of combining these two to deepen our understanding of cardiovascular pathologies. On the mechanics side, I combined mechanical testing of both human and mouse arteries with microstructural quantifications and theoretical modeling to get an understanding of the local function of arteries. And this was paired on the global side with a combination of ultrasound, CT, and catheter or tail cuff measurements of blood pressure to get an idea of what are the boundary conditions to which these arteries are exposed to. And so by spearheading these methods, together with our collaborators, we combined them into sophisticated computational models that allow us to simulate the entire vascular tree, at least the central vasculature. And we addressed several questions with this combined computational experimental framework. And we performed several studies, but at least related to the issue between arterial stiffness and pressure, if I need to sum it up into one statement, I will conclude that loss of functional elastin stiffens arteries, which then increases blood pressure and alters wave propagation. So I just wanted to show this because this is pretty much part of my expertise, and it's something that I would like to pursue in the future. But I would like to dedicate the rest of today's talk to what is the effect of mechanics on tumor evolution. Because while it is known that the role of mechanical forces is well established in the cardiovascular system, their role in tumor progression is still a matter of debate and active investigation. What we know is that tumors are highly heterogeneous tissues. Here you can see a multi-photon image of a human tumor where cancer cells are labeled in green, here in the bottom right, in bottom left, and the capsule rich of collagen that surrounds the tumor is labeled in red. Now, in addition to being heterogeneous, tumors are actually highly dynamic. Part of that dynamic takes place at the invasive front, where cancer cells, either individually or as cell clusters, invade through the tumor capsule that is rich in collagen and stromal cells. But also the tumor core, despite being densely packed with cancer cells and low in collagen, is highly dynamic. And in fact, recent evidence shows that cancer cells in the tumor core can display highly coordinated motions. Now, one of the questions that I'm interested in addressing is how the bidirectional interactions between a tumor and the ECM govern invasion. So one of my first questions was to explain how the dense capsule of collagen that surrounds the tumor is formed. Now, to do that, I used a simplified system, a 3D breast cancer spheroid system that is shown here on the left, where cell nuclei are labeled in red, whereas collagen is labeled in green. Now, remarkably, this simple system can show the same patterns of collagen remodeling that are shown in vivo, with a locally dense capsule of collagen formed at the spheroid boundary. Now, going into this, my hypothesis was that the forces generated by tumor growth will compress collagen locally and will collapse collagen to generate this remodeled collagen capsule. To do this, I performed several experiments. There's no need to go into the detail, but ultimately, all of this was summarized by a computational model. Here you can see a short video. And again, details are not important, but since I'm teaching statics this semester, I would like the students in the audience to know that this is just a glorified version of Newton's second law, F equal ma. And so thanks to this model and the experiments that informed it, we were able to reproduce, at least in terms of localized deformation, results that are similar to our experimental observations. But in addition to that, the advantage of such models is that allows us to gain insight of what are the mechanisms behind this remodeling phenotype. And we found that fiber buckling, that is the fact that fibers collapse under compression, and cross-link capture, that is the fact that fibers permanently detach from each other under the action of forces, are underlying this remodeling phenotype that is well documented in tumors. Now, while this study was very insightful, it did not really address how the ECM density regulates invasion. And so to address this question, I started looking at different conditions. So I monitored tumor invasion by varying the degree of ECM confinement, that is by changing collagen concentration, and by changing the invasive potential, that is by using different breast cancer cell lines. By doing so, I observed a variety of invasive phenotypes, including no migration, collective cell migration, and single cell migration. So at this point, I was looking for a unified framework that will allow me to explain such diversity in cancer cell behavior. And while I was doing that, Jeff Fredberg at Harvard suggested that we look at this problem as a phase separation problem. That is, starting from a solid tumor, then the various conditions can lead to different migratory modes via separation into solid-like, liquid-like, or gas-like material phases. Now, because of the theoretical background of this, to confirm that our hunch was correct, we needed to quantify cell shape, as you can see here, and cell dynamics. And both these measurements supported the hypothesis that a non-equilibrium phase separation process was underlying tumor invasion. Now, there's no need to go into the detail of the further experiments, but we performed them, and ultimately, we developed a computational model in which we tuned cell motility and collagen density while leaving fixed all the remaining parameters. Much to our surprise, we were able to recapitulate all our experimental observation. So overall, these results indicate that a non-equilibrium phase separation process represents a new framework to explore tumor heterogeneity and invasion. So at this point, I'd like to take to pause for a moment and put these findings in context. We have long known that the biological state of a tumor controls invasion. What I just showed you instead supports the notion that both cell and matrix properties contribute to determine the material state. And the material state of a tumor is what governs invasion. It is still very unclear how the local material phases impact the biological state. And this is going to be very much a matter of investigation in the future. So in my last slide, I would just like to show you how I started tackling this effect this year of how the material state affects energy metabolism. So the question that I posed is, what changes in cellular energetics occur during collective migration when cells become more liquid-like? Now, to do this, I reduced the dimensionality of the system. I went from 3D to 2D. Here you can see we culture cells in this rectangular area, which is surrounded by a PDMS barrier. So when the epithelial sheet grows, it will be limited by the PDMS barrier. At that point, we can simply peel off the PDMS barrier, and what we observe is the following. Cells will start migrating collectively on either side. And we can clearly identify a non-migratory interior that is solid-like and a migratory front that is liquid-like. Now, to solve this problem, I teamed up with Steve DeCamp, and we currently have a paper under review, who is an expert in measuring spatio-temporal patterns in cell morphology and mechanics. So as you can see here, there are measurements of a variety of markers during this process of collective migration. And what both Steve and I were interested in doing is to measure metabolism at the same spatial resolution, so not in cumulative. And so we use various methods, and in particular, I would like to highlight fluorescent lifetime of the metabolic cofactor and ADH, which is something that I'm very much interested in using in the future. And overall, we found that collective migration shifts cell metabolism towards glycolysis. So I would like to just conclude with stating the mission of my lab, which is to advance basic understanding of tissue mechanics and remodelings and the role it plays in disease initiation and progression. And I hope that I showed you that my lab has complementary expertise in biomechanics, biophysics, and mechanobiology to accomplish this mission. Not only that, we'll need collaborations and active efforts into imaging, biology, and theoretical and computational modeling at different length scales so that we can have a multidisciplinary and multi-scale approach that can facilitate in collaboration with other groups. And so with that, I'll just finish by acknowledging NIH for funding, and I would really love to hear any comments or any question that you might have here, or you can just email me. Thank you.
Hello. Can you see my slides now?
Yes.
Awesome. Okay, I'm very excited to present my research, and I'm hoping to pick up a lot of collaborators and students and postdocs. So my lecture today will be on the microfluidic platforms that I develop in my lab to quantify immune cell decision-making phenotypes. So the reasons why that University of Texas at Dallas is the perfect fit for my lab, and I love that I pulled this picture from the internet, but this is actually the view from my office, is that it's a great combination of engineering, biomedical engineering, bioengineering, and as well as electrical engineering. The Texas Instruments developed a nice clean room here, and I can make my microfluidic devices using that resource, as well as the collaboration and proximity to UT Southwestern Medical Center, where I can get patient samples and use my enabling tools to measure immune cell phenotype with that engineering tool set. And so I just wanted to put this at the beginning of my talk that I am looking to hire several graduate students, undergraduates, and a postdoc. So please email me at caroline.jones@utdallas.edu if you have any questions or are looking for a job. So the Jones Laboratory of Immunoengineering and Therapeutics focuses on three different main aspects of immunology and engineering. We first look at technological innovation, and we engineer innovative lab-on-chip platforms to model and study immune cell function and phenotypes. And then we also use those tools to investigate the biology of inflammation. So specifically looking at epigenetic reprogramming of innate immune cells. And then finally, we like to take these tools that we engineer on the UT Dallas campus and use them to measure septic patient immune signatures at UT Southwestern. So to take a step back and give you the big broad picture of what I do in my lab, everyone is really familiar with the routine blood tests. So for instance, getting a chemistry panel of your cholesterol levels or your glucose status. There are more advanced screens now looking at inflammation levels, for instance, C-reactive protein, or just running a blood count or a CBC on platelets, red blood cells, and white blood cells. So in my lab, we focus on white blood cells, and we know that just looking at the counts of these white blood cells is not enough. And we really need to get the how these cells are functioning and use tools to be able to measure phenotypes and not just counts. And I don't really need to tell you why this is important. That's the reason why we're meeting in this virtual format, is because of COVID-19. And so understanding immune function and being able to design immunotherapies to be able to improve the immune system and reduce inflammation is extremely important. My lab focuses on sepsis, and that is the overreaction of the immune system. But we also apply these same tools to investigate cancer and potentially in the future, COVID-19. So the tools that I make in my lab, we look at cell migration and how cells are able to fight pathogens. And I'll give you just a quick overview. Normally, biologists, if they were looking at cell migration, would just use a Boyden chamber where you put cells in the top of a chamber and then a signal or a chemoattractant in the bottom of the chamber and just count at a bulk sample how many cells migrate. But you lose a lot of information here. And so when you develop microfluidic tools where we can have these well-defined mechanical channels and chemical gradients, we can look at how the cells migrate and interact with pathogens at a single cell level. And that way we can look at the heterogeneity in cells. We can get a lot more detailed information. So we can look at even velocities of cells, decision-making of cells. If you see this maze here, you can see how the cells are able to actually navigate a maze even without a chemoattractant signal. And we can make much more biomimetic situations and microenvironments for the cells. So here, hopefully this movie is playing for you. You can see on the left, this is one of the small microfluidic devices. And oftentimes you'll see in lab on chips that's next to a penny. It's a very small device. And this is great because we can look at many, thousands and thousands of cells on chip and be able to measure their phenotypes, even from just one droplet of blood. So here you can see that there's a red, a yellow, and a blue color kind of going out of these tubes. So we were able to measure 3 different chemoattractants. And then in the central picture, you see these are my neutrophils migrating to a pro-inflammatory signal. And you can see that they're able to really quickly migrate through these mazes and migrate to the signal that they're supposed to. And one of the things about making these devices is that we make all of the devices in a clean room. So here's a picture of the clean room that I worked in when I was a postdoc at Mass General Hospital. And a lot of times when people see us in these suits, they assume that we're working with BSL-3 infectious agent and that we're wearing all this protective gear for that reason. But actually, it's the opposite. We're trying to protect our devices from any piece of dust or hair because these channels here where you see these neutrophils migrating through are about 6 microns in diameter, so less than a width of a hair. And so any type of dust particle could interfere with our chip. And what we do to make these movies of cells is that we use time-lapse imaging. So we're able to take pictures of each position on the chip and then put together every 2 minutes and then put together how these cells are actually moving. So the reason why I study sepsis is that it's the leading cause of deaths in hospitals and intensive care units. Even in this COVID pandemic, you can see that the cytokine storm or hyperinflammatory response of the later stage of the disease is very similar to sepsis, and many of these patients are categorized as septic. It's the life-threatening response of the body's immune system to an infection, and that can potentially lead to tissue damage, organ failure, and death. So these cells, instead of going to fight the pathogen or the infection, will migrate to a healthy organ or a healthy tissue and cause collateral damage to the lung or heart. And so the main focus of my lab is to ask very basic questions. For instance, how does an individual white blood cell, also called leukocyte, prioritize which chemoattractant gradients to follow in a complex tissue microenvironment? We know that they're around more than one signal, so how do they prioritize that? And is there some type of hierarchy? And then is the leukocyte's compass a microbial killing mechanism set by prior inflammatory or pro-resolution signals? So right now I'm teaching immunoengineering for the students out there in my class, and I tell you that the innate immune system does not have a memory and the adaptive immune system does. But as we look at more complexities, we're realizing that epigenetic changes can lead to memory even in the innate immune cells. So meaning that the microenvironment that innate immune cell was exposed to will determine its subsequent phenotype upon infection. So one of the first projects that I'm starting up here that I began during my postdoc, continued at Virginia Tech, and now will bring to UT Dallas is that I measure the innate immune cell function from septic patients. So here it's great. I can collect the blood sample right at UT Southwestern, and then have a microscope right on the UT Southwestern campus and isolate neutrophils from, and I look at neutrophils, that's the main white blood cell. So I isolate neutrophils from the septic patient, and then I can run a migration assay. And during my postdoc, I was able to identify specific phenotypes that were novel or only identified in septic patients versus healthy donors. And so using this, I was able to score the phenotypes of these cells and diagnose septic patients from just the phenotype of their neutrophils or innate white blood cells. And what's really cool about these studies is that I was able to identify a specific phenotype even the draw before the sepsis diagnosis in the hospital setting. And so this could be a good marker, so monitoring for early warning for sepsis. And then ideally, and in my subsequent studies, I want to understand the biological mechanisms and create more sophisticated tools to be able to probe some of these phenotypes to understand how we can prevent sepsis. So that leads into my time at Virginia Tech. I developed a second iteration of this device design. As opposed to having cells that are migrating just to one signal, I actually put cells in a channel and in between two different signals. And I won't go into all the details here because of the time constraints, but if you want to chat about this later, it's a really cool concept that priming with different inflammatory levels will change the phenotype of cells subsequently to an infection. And to compare that to the COVID-19 pandemic, you hear a lot about pre-existing conditions or age determining the outcome or having a huge influence on the outcome of the SARS-CoV-2 infection. Well, here you see that if you have a super low dose of LPS or very low levels of inflammation, that can precondition the cells to respond in an incorrect way. And so for simplicity's sake, let's say that the green signal is a good signal. That's where the cells migrate correctly to the microbial infection, and the red signal is a bad signal, saying that the cells migrate to inflammation. And we showed with this paper that you can actually tune which direction the cells will go based off of their previous microenvironment. And so I do work in this interdisciplinary field, and so not only do I work with biologists and engineers and physicians, but I also work with mathematicians because we get so much data from each of these experiments that we have to model and understand, the basic mechanisms of why the cells are making these decisions. And so a lot of this work was done by Dr. Brittanyboribong. She was my first graduate student who graduated, and now she is a postdoc at school. So here are some of the movies that she did. Sorry, let me go back. Sorry, I want to be able to play these movies. Oh, here you go. So here you can see some of these movies where you have the blue cells in the central channel, and you can see that cells are going towards both signals. And so this is just healthy neutrophils. This is modeling a healthy response. And then with super low dose LPS, you can see that many, many more of the cells are migrating towards the signal. And so from this, you can really see that we are able to take cells that are isolated from human samples and then treat them in certain ways and then look at how these cells respond. So I won't go into all the details of this experiment, but we were able to show that if we stimulate these cells with a low dose LPS, that we can alter the decision making. So we can push the cells to go more towards the inflammatory side than the resolving side, and that may model some of these low levels of inflammation. And then we put that into a modeling cycle where we looked at specific receptors and different priming, either with pro-inflammatory signaling or anti-inflammatory signaling. And we're able to model some of these phenotypes based off of the receptors and kinases that we saw in the cells. And so we are able to not only measure the migratory phenotype, we're also able to take these small numbers of cells off chip and then probe them using ddPCR to look at their proteins and receptor levels. And then I have to show this. I'm glad that Jacob showed his study on elastase in the microenvironment, but we're not only showing migration, but we can also show elastase release. So elastase cleaves elastin in the microenvironment. And so you can see here, this is a droplet of neutrophils, and they're very, very pro-inflammatory. They're producing a lot of elastase, and our biosensor shows this great bright green signal. And so that would remodel the microenvironment, making it really pro-inflammatory and damage tissue. And so we're able to do that with on-chip, these on-chip biosensors. And here are macrophages migrating from the blood sample. And you can see that they produce a lot less elastase, and that there's even heterogeneity in this one sample, showing that the M1 versus
Caroline,
M2.
I don't know if it's just me or anyone else, but my my screen isn't updating. It just shows like the movie before you started it.
Okay, let me see.
Yes, thank you, Heather. Yeah, I think your slide might not have advanced, and we are wrapping up the time that we have for your presentation. So do you want to give us a conclusion? And I'm sure that folks who are interested can contact you and continue the conversation. And of course, we'll go to questions here in a few minutes after Dr. Obaid.
Okay, sorry. Okay, so then I'll just have 2 more slides. And so we have those studies where we're showing immune cell function, but then we also can design hydrogels on chip where we can culture spheroids and then look at using RNA-seq to measure the cancer phenotypes. We can also integrate biosensors there, measuring specific receptors on the cell surface. And then finally, we can not only measure cell migration, but then we can actually try to control and tune cell migration. So here's a paper we just published last month on Lab on a Chip, where we can induce electric fields to drive drug delivery as well as cell migration on this similar microfluidic platform. And then finally, we are making some outreach programs where we can actually, and a lot of people are learning, students are learning online, where we can do virtual reality and actually see some of these, learn about immunology and experience how it would be to be an immune cell fighting a pathogen by putting on some of these virtual reality goggles and seeing it from the viewpoint of a cell. So with that, I will conclude. I have recruited 2 students so far at UT. And I'm looking to recruit several more and a postdoc, and undergrads are always welcome. So please email me. And sorry for going over.
Thank you so much. Dr. Obaid.
Hi. Okay, so my name is Girgis Obaid. It's a pleasure to be here today, and I'm going to talk to you about the program that I've recently started this year at UT Dallas Bioengineering. My research is focused on optically active nanotherapies, and these are effectively traceable and personalizable in terms of their nature, and we develop these approaches for cancer management. So before I go on, I want to introduce the team. I've been really fortunate to find some fantastic members in the team so far that have joined. So the program itself is called Molecular Imaging and Optical Nanotherapeutics, MION for short, and we are based at the BSB East Wing on the 2nd floor. 12.460 is the general lab space. So I myself have a background in biochemistry and chemistry, and I trained at the Wellman Center for Photomedicine for my postdoc. So that's anything and everything to do with light-activated treatment. We also have Dr. Navdeep Srivastava, who's an expert in luminescent and magnetic nanomaterials. We have an undergrad student, Meena, who's he's got a double major in biology and healthcare management. We have Chanda Bandari, who's a PhD student. We have Nimit, who's also a PhD student, and Sushant, who's a PhD student. Unfortunately, all 3 PhD students are stuck outside the country because of COVID, but fingers crossed they'll be able to join us soon. And so an overview of the research program itself. It effectively focuses on a very cross-disciplinary area of research, and it's synergizing molecular imaging with optical nanotherapeutics. So these are light-activated, nano-sized materials that are used as effectively cancer drugs. And we range in terms of the expertise from photochemistry all the way up to tumor biology, immunotherapy, nanotechnology, and of course some imaging. And so a few of the concepts that we bring in together in the program include photodynamic therapy, which is a light-activated cancer treatment, and that's effectively right at the core of our research program. We also then have some technical issues with the, but nonetheless, so we have precision medicine being one of the focuses as well. How do we tailor our treatments to specific patients and to specific tumor types? And yeah, I do apologize, this has been glitching multiple times, but nonetheless, we also focus on nanomedicines and drug delivery. We look at image-guided nanoparticle design. So we use molecular imaging as a way to design nanoparticles. I'll talk a little bit about that later. And ultimately, we use rational combinations of anti-cancer treatment modalities in order to enhance the efficacy and reduce the toxicity for patients. And that's really the goal there. So just introducing photodynamic therapy to those who are not familiar with the concept. It involves first the administration of what's known as a photosensitizer. That's a light activatable drug, and it accumulates in the tumor after it's distributed throughout the body. And once it's accumulated, we then have activation of the sensitizer in the specific tumor to elicit photodestruction. So we're actually destroying the tumor tissue and a myriad of other secondary effects, which I'll touch on a little bit later. But ultimately, the real power of photodynamic therapy, also known as PDT, is that it's a highly controlled modality, and it generates these reactive molecular species with specific control over the time of when the treatment is induced, the activation of where the treatment is going to occur, and the dose of how much we're going to deposit in the tumor. And so a lot of the work that I've done before is, as I mentioned with the nanotechnologies, it includes various different formulations of nanoparticles and targeted nanoparticles. And my specific focus is on precision medicine. So how do we tailor these nanoparticles specifically to tumor types? And that's facilitated for us by molecular imaging directed system design, and then ultimately the synthesis based on that design. So molecular imaging for us is incredibly important in that regard. And so photodynamic therapy, I mentioned, has a myriad of these secondary effects, and that's also incredibly important in terms of being able to evaluate what they're doing for patients. So in a collaboration with Dr. Charles Lin at Mass General also, we looked at stromal modulation. So this is a paper we're publishing soon, but we use second harmonic generation imaging of tumor collagen specifically to look at how the collagen density and the collagen order changes in response to photodynamic therapy. And so we can generate these second harmonic generation signals, and the one on the right here, what's known as TIPMIL, this is effectively the most potent nanoconstruct that we designed. And we show that with imaging, it is also the most potent in terms of its ability to modulate the collagen in the tumor. And for us, that means that it's remediating desmoplasia. And desmoplasia is effectively fibrosis that we see in tumors that leads to many problems with treatment resistance and drug delivery. So this is quite an interesting angle with regard to photodynamic therapy. There's also something that we're interested in, which is the immunostimulation and immunotherapy component. So photodynamic therapy itself triggers vast combination of different immunological responses, whether it be cytokines and immunogenic cell death. And so the immune system plays a massive role in the response to PDT in of itself. And a few different immunomodulatory effects include infiltration of leukocytes or blood cells, or immune cells rather, into the tumor. We've got repolarization of tumor-associated macrophages and cold to hot transformation of tumors, and that's now being leveraged in the field for combining PDT with immunotherapy. So this is a particular direction we're also interested in going. Now, the synergy between molecular imaging and optical nanotherapeutics is really the niche of what we are going to be working on in our lab specifically. So just to give you an example, these nanotherapeutics that we design are really rather complicated, both in terms of their composition and in terms of their stability and effect. And so when we take molecular imaging, we can get a whole load of information on the functionality of how these nanotherapeutics perform in vivo. We can probe what are known as nano-bio interactions. So how's the nano construct interacting with the body? We can look at personalization of these nanotherapeutics, and we use molecular imaging to guide that. And we can also we can also evaluate treatment response and thus optimize dosimetry using molecular imaging as well. And then we can trace and track the induction of these secondary responses to therapy. And so we have a number of emerging collaborations that we're really quite excited about. The first I'm going to mention here is with Dr. Kenneth Hoyt's team. And so Lokesh and Junji have been fantastic help in starting this collaboration. We've got a few experiments going. And we're going to be using a technology that's been developed by Dr. Kenneth Hoyt, known as quantitative photoacoustic imaging, to get cellular information on how the vascular effects of photodynamic therapy will improve the delivery of nanomedicines into tumors. And so we're designing this construct that's targeted to the tumor vasculature through these antibodies and has a photodynamic agent on one end and a photoacoustic agent on the other. And so we have some preliminary data here that shows that using a vessel phantom, we can actually visualize a precursor of this construct, and we can actually see this construct in the blood vessels. This here is the ear of the mouse as a 3D reconstruction. Another collaboration which we've had for a couple of years now that we're applying for grants with is with Dr. David Kessel at Wayne State and Dr. Imran Rizvi at UNC Chapel Hill. And here we're leveraging the subcellular precision of nanoformulations. So we direct them to specific sites inside the cell to have a control now over which death modality we're going to be triggering. And because of that, we can then combine these concepts with various different treatment modalities such as chemotherapy and radiation therapy. And this becomes helpful for treatment-resistant tumors. Another collaboration, which we're really excited about, is with UT Southwestern Radiation Oncology, Dr. Saha and Dr. Story. We're looking now at the synergy between photodynamic therapy and radiation therapy, specifically for head and neck cancer. And the idea is because they are two non-overlapping modes of tumor killing, we can circumvent issues with radiation resistance in the tumor for head and neck cancer specifically. And beyond that, we also have a number of exciting discussions with UT Arlington faculty. And within UTD, we've got Dr. Circe Blaris-Fay and Dr. Mishra, who's our next door neighbor. And these hopefully will bring some fruitful collaborations in future. And so I want to just say a special thanks to all of the UT Dallas faculty, especially those at Bioengineering, who've been incredible support, as well as the staff for helping getting our lab up and running. And we're really excited to move forward. So if anybody wants to reach out, my contact details are here on the right. So thank you.
Thank you very much. And I think all of you have heard the brilliant presentations of all our four faculty. So we'll open this up for questions. So please use the raise hand function. I'll try to call out the students first. So please go ahead.
Awesome. I had a quick question for Dr. Ferruzzi. I hope I pronounced that correctly as well.
Yes, you did.
Awesome. I'm a new PhD student working in Dr. Meyer's, or Dr. Hayenga's lab with Dr. Meyer as well. We're working with biomechanics and mechanobiology as well. And I kind of just had a quick question. You were talking a little bit about the, how you were studying the arterial stiffness and blood pressure in a mouse aorta. And I was just wondering what kind of imaging techniques y'all were using to kind of get that three-dimensional model of the artery.
That's a good question. So I guess if you're referring to the model that I showed in the center of the slide, that was something those were micro CT imaging that I collected back at Yale. And then obviously those were segmented and used to generate a fluid structure interaction model. But the raw data were CT, micro CT.
Excellent. Okay, thank you very much.
I think there's a question on the chat, right? So what skill sets do researchers look for in undergraduates for consideration?
Do any of our speakers want to weigh in on that? What kind of skill sets you're looking for in undergraduate students?
I would say it's not necessarily a skill set, but a passion for that type of research, because any of the skill sets I can teach you in my lab. So it's less of a skill set and more of a mentality and really passion for the research.
I guess that I think all of us would like to echo that. It's more of a passion. I think especially experimentally, we're always very happy to train students. I guess that from my perspective, a basic understanding of regular statics, which I'm teaching, or some basic biomechanics, which I know is also part of the offering of the department, it will be will facilitate your integration in the lab.
Yeah, I agree also with what Caroline said about the passion. At least on my end, some experience of knowing how to work in a wet lab. So vague things like pipetting and stuff can be helpful as opposed to purely theoretical expertise. But nonetheless, these are all completely trainable, like Caroline or Dr. Jones said. So I agree.
Yeah, I agree with all of you.
Yes, hi. And thank you for all of your presentations. They were really great. So I have a question for Yichen about PCA. So I'm wondering how many significant components you usually get for example, modeling heart tissue. I know it's going to be different from one case to another case, but I just want to know the range. How many components do you usually get?
Yeah, actually, that's a great question. That depends, to be honest. So when we perform PCA, we have quite a lot of coefficients out. So generally, we will get maybe 10 to 20% of those coefficients. But if they are too much, maybe we'll just try to reduce it to 5%. That depends on different applications.
I see. Thank you.
Yeah, thank you.
Hi, this is John Hansen. Sorry, I wasn't able to see all of the presentations because I had another meeting, but I wanted to say ones I saw I was really impressed with and very happy to see our young faculty's diversity and strength in their research. I'd like to maybe make a comment. If you check in the chat, we do have a very active program for Johnson School Undergraduate Research Awards, and we've had many undergraduate students in bioengineering that have had very successful engagements with our faculty in bioengineering. So if you have an interest in working with any of these young, energetic new faculty members, as well as those faculty members that are already in our bioengineering department, I don't want to say not as young, but maybe young, you'll find that very engaging, and I think you'll see that it's a valuable experience. I know faculty may already have research funding, but there's a little extra funds there that go directly to the student, but it's also something that's nice to add into your resume, especially if you're looking at going on to grad school at some point in your careers. So thanks.
Thank you, John, for that. Okay, so Heather, go ahead.
Thank you all. I know these were just flash lightning talks, and I know there's much studies and experiments that were done to get to those results. But my question is for Jacopo Ferruzzi. Can you explain, maybe you showed really nice single cell versus group cell migration in cancer. What are some of the mechanisms behind that? What's like mechanistically driving the mechanotransduction, or like the pathways, or just comment on that more?
Absolutely. Well, I guess that what Heather is referring to is probably the biological mechanisms of collective migration as opposed to single cell migration. And in there is a whole plethora of work regarding also epithelial to mesenchymal transition. So in terms of mechanisms, it is an actually very complicated process. So instead of looking at that particular mechanism, we looked at an integrative framework, something that will allow us, at least physically, to take cells with different epithelial versus mesenchymal patterns or different ECM environments, and sort of be able to at least identify what are the different phenotypes and what drives those phenotypes. Now, mechanistically, what I can tell you is that there are this fluidization of tissues is associated through RNA-seq, for instance, like Caroline was mentioning. It is shown to different pathways that are activated there. So this is still much a matter of investigation. So long story short, I don't know exactly what is driving those, but this is an interesting challenge for the future.
Thank you.
I think we have a question from Jude Franklin.
Yes, sorry. I have a question for Dr. Obaid in general. So just comparing your research to other standards of care that exist out there, what makes Like I'm sure there's several other things that are for precise cancer treatments that are out there, like modifying T cells and things like that. Well, why do you think this is more, I don't know, maybe has a better efficacy or a long-term effect? Why do you think this would have better results versus the things that already exist for these treatments?
That's a really good question. I think to summarize, it all comes down to the fact that it's a multimodal treatment in of itself. And so it can't be considered purely as just a T cell activating treatment. It can't be considered just as a stromal modulating treatment. It's the idea that it does so many of these together and that you can control what it is that you're doing with it based on the parameters that you are allowed to. So for example, the light dose, how much light you're shining, what the energy is of that light that you're exposing the tissue to. So you have so much control. And even though it's one specific quote unquote treatment modality, there's so many multimodal features to it that you can control. And I think that's really what makes it in terms of being unique compared to other treatments. But again, this really can go into a lot of detail. So I'm happy to talk offline too if you'd like to.
Sure, I would love that. Thank you.
Oh, I guess I could ask another question.
Sure.
Really quickly, I was wondering what you guys are all looking for in a collaborator. I mean, that's a really open-ended question. So, are there skill sets like to complement yours, or just any kind of, just in general, what you're looking for?
So I can start just to briefly mention. The directions that we go can be so diverse, and so it's not necessarily about finding somebody with a specific skill set, even though of course that definitely helps. But the idea is that can we work with that person, and do they have the same kind of vision? Do they share the vision with us in terms of where we want to head with the research? So we take the research in so many different directions that it almost doesn't matter at this stage. It's more about the enthusiasm and the excitement. So that's it from my end.
Yeah, I think we can all agree with what Girgis said in terms of enthusiasm and attitude. Definitely complementary expertise and more clinical translation. It's definitely what I'm also looking into it, but really the attitude and the excitement and the vision is the key factor there.
I'm also looking for clinical translation as well as some in vivo imaging techniques that I can match up with my microfluidic model. So if I, using a murine model or other animal model, can I ask some of the similar questions using hydrogels and match up the phenotypes that I see with microfluidic devices? But I also agree with just being able to work well with a person and having a collaborative environment for a student to learn many more skill sets would be amazing.
Yeah, actually, I'm open to any kinds of collaboration because they have so many directions. But I want we can support each other because we have so many directions for different purpose, different applications, different techniques. So it's not a specific collaboration. Thank you.
So that's all I think we have in terms of questions for right now. So I wanted to tell you all, I might be freezing, okay, but I wanted to tell you all that at its peak, we had 85 attendees. So clearly lots of interest in your work. And all of you who are on the call still, please reach out to them. We are looking to build collaborations, expand our reach, and we will make this available through our department YouTube channel, their talk, and we can always share contact information. Thank you all for attending. Thank you, the four of you. Really appreciate you taking the time to give a quick preview. It's a good research shot. Thank you so much and have a good weekend.